noble gas-protein interactions through thermodynamic analysis.
Hi, I'm Alan, and I work with my mentor Nancy Wiebelhaus at the Fitzgerald Lab at Duke University. And my project was on identifying noble gas-protein interactions through thermodynamic analysis. So the noble gases are a family of elements that are often found in their gaseous state. They are chemically inert and unstable, and as a result, we don't often see them interacting with biological systems. However, we are interested in these noble gases for two reasons. First, when there's nuclear activity, radioactive isotopes of certain noble gases are produced as a result, and the Defense Threat Reduction Agency has given my lab a grant to explore using biochemical means to identify and track these radioactive isotopes. Additionally, certain noble gases such as xenon are known to be extremely potent anesthetics. Xenon, for example, is a very potent anesthetic, and due to its, since it is very safe, it's not known to behave toxically or reactively with the body. It is a good candidate to explore the mechanisms and behind anesthesia in biological systems. So the objective of my project is to develop a model experiment that can be used to identify noble gas protein interactions. So my experiment is based on this theory of the structural stability of proteins. So proteins are a string of amino acids, and they will fold together into certain shapes, and based on its shape, it will determine how the protein functions. And when this folded protein, the folded protein is how proteins usually exist in its wild type or natural state. And when this folded protein is exposed to high pHs, certain temperatures, or certain chemicals, the protein will undergo a process called denaturing, where it will unfold. And the point at which a protein will resist unfolding is determined as the protein's stability. So if the protein's stability changes, it means that the protein is interacting with another substance or ligand. So for example, in this previous study, they looked at lysozyme and its interaction with this drug. So in this graph, we basically have the concentration of a chemical denaturant in the x-axis and how much the protein has denatured on the y-axis. So if we look at the black control curve, we can see there's kind of these two states where it begins at a folded state and eventually goes towards an unfolded state, and it will undergo this very quick reactive denaturing in the middle. And when lysozyme interacts with this other chemical, it will shift to the right, so it will unfold at a higher concentration of denaturing. So another study that has used theory to identify drug interactions is this study with mannitidine A. So mannitidine A is a natural product that has been shown to have anti-cancer activities, except they didn't know why it had anti-cancer activities. So what they did was they took a cancer cell, they split it up into all of its different proteins, and then they exposed all the proteins to mannitidine A. And then they isolated 5 or 6 proteins that interacted with mannitidine A, and that gave them an idea of how mannitidine A gives anti-cancer activities in cells. So in my project, I use myoglobin and xenon gas as my model protein and noble gas. I chose myoglobin because it is a very well-studied protein in its form and function. Globin proteins like myoglobin have the function of transporting or holding gases through the body, and that might explain a mechanism of how xenon diffuses through the body. And also myoglobin has already been shown to bind with xenon gas. So the first way we can track the stability or the structural change of myoglobin is through a UV visible spectroscopy experiment. So myoglobin is a globin protein, which means it has this iron heme group in its center. So this heme group usually gives off a very red color, which is what gives hemoglobin, myoglobin its red color in muscles or blood. And when this myoglobin unfolds, the heme group inside of the protein will be exposed to outside solvent, and because it interacts with solvent, it will change color. So for example, this is wild-type myoglobin, and when we denature it, it will turn this lighter orange color. And what can happen is we can track this color change using UV visible spectroscopy. However, UV visible spectroscopy can only be used with globin proteins. So to expand our field of proteins that we can use, we also use a mass spectrometry-based strategy. So myoglobin has 3 amino acids called tryptophan within its structure. And since tryptophan is hydrophobic, it is often found on the inside structure of proteins. So usually when the protein is folded, it doesn't interact with the stuff around it. However, when we denature myoglobin and the tryptophan is exposed, we can introduce a chemical called HNSP. And the HNSP will attach itself to tryptophan and add a mass of 58 Daltons. So using mass spectrometry, we will measure the mass of the protein, and if it changes, or when the mass changes, it will indicate when the protein is unfolding. And so, yeah, we use mass spectrometry. And so basically how our experiment works is we prepare the protein, one prepared under xenon gas or one under our control gas. And for our control, we just use house air, what we're breathing in right now. And then we will expose it to a concentration of guanidinium chloride, which is what we use as our chemical denaturant. And then depending on the method, we would find its stability at each concentration of chemical denaturant using UV-vis spectroscopy or mass spectrometry. And then we could build a fraction not denatured versus concentration curve based on that data. So first, from our UV visible spectroscopy data, so we have our concentration of denaturant on the x-axis and the amount of protein that has been denatured on the y-axis. And it has the characteristic S shape that we were looking for. And so with our xenon and control curve, we see that the xenon curve shifts to the left by about 0.148315. So this C1 half value here is basically determines at what concentration of chemical denaturant will half the protein be unfolded. And then with our mass spec data, I wasn't able to build a xenon curve yet, but we still have a similar S-shaped curve with our control. So what we found interesting was that according to our data, it suggests that xenon destabilizes myoglobin structure since it unfolds at an earlier concentration. If you can see, our change in C half value is about 0.148315. So at my lab, we do a lot of this type of research, and our threshold for a hit is a change of at least 0.1. So this gives us enough to continue looking at this xenon-based structural research. Yeah, and in the future, we also want to hopefully see that with the mass spec experiment as well, we'll see a similar destabilizing effect of xenon. So just for a quick summary, we can use our end goal is to use mass spec to analyze proteins. UV-vis was our original idea because it is very cheap and it is very easy and quick to do. But mass spec offers a lot more different proteins that we could look at. And one thing we want to do in the future is also control how much gas that we expose to the proteins. So what we did in this experiment was that we just flowed xenon gas through a buffer solution for a set amount of time and a set pressure. But hopefully we are currently experimenting using gas chromatography to find the exact amount of noble gas that is in our solution. So for our take-home slide, eventually we hope to use this experiment to find more proteins that can interact with noble gases. And one thing we could also do is we could take a neural cell in the future, split it up into those proteins, and see if any of those proteins will interact with xenon gas as a way to suggest how xenon has anesthetic behavior. And we can also use the experiment to build proteins that can hopefully interact with radioactive isotopes of noble gases. So thank you to NCSSM, my mentors, and my mentorship teachers. And any questions?